Actin filament assembly promotes the formation of intercellular adherens junctions, and it's well known that the actin-nucleating ARP2-3 complex and Rho GTPases such as Rac1 regulate actin dynamics at nascent cell-cell contacts. The role of actin-nucleating Formin proteins is less well understood, however, something that Robert Grosse from the University of Marburg in Germany wanted to address in epithelial cells cultured in a physiological 3D environment.
So one key aspect that we are interested in my lab is trying to understand actin events as they happen in real time in living cells. And so we had this idea that we wanted to look at what happens during the birth of the adherens junction in healthy epithelial cells that are cultured in the 3D environment.
Grosse and colleagues, led by Katharina Grikscheit, studied junctional actin assembly in breast epithelial cells, which, when cultured in 3D matrigel, initially divide to form two daughter cells that come together to form a new intercellular adhesion. The cells then gradually develop into large spheroids containing a central lumen. The Formin protein, Formin-like 2 or FMNL2, was highly expressed in these cells and localized to nascent cell-cell contacts, so Grikscheit et al. Examined the effects of depleting this protein on junctional actin assembly.
And so we use this actin reporter, LiveAct, to look at that event. And you can see a very nice labeling of LiveAct at this dual cell spheroid. And when we silence FMNL2, this nice F-actin structure was interrupted. It was not intact.
Photobleaching experiments indicated that the amount of free, non-polymerized actin at cell-cell contacts was increased in the absence of FMNL2.
And it was really specific for this junctional pool of GFP actin that was detectable at the cell-cell contact site, but not at the cell cortex somewhere else or in the cytoplasm.
Junctional actin assembly was rescued by a constitutively active version of FMNL2 lacking its autoinhibitory DAD domain, whereas knocking down other formins had no effect on actin formation at cell-cell contacts. Over longer periods, FMNL2 depletion impaired the spheroid's ability to form differentiated lumens, though whether this phenotype is due to defects in junctional actin remains unclear. Grikscheit et al. Then turned their attention to how FMNL2 localizes to cadherin and catenin-based adhesions.
We did biochemical experiments and live cell experiments, and what we observe is that there seems to be a dynamic RAC-dependent association of FMNL2 with the ecadherin alpha-catenin complex.
Rac1 is a key regulator of junctional actin, and while a constitutively active version of the GTPase promoted FMNL2's localization to cell-cell contacts, knocking down Rac1 decreased both FMNL2 recruitment and local actin assembly. This phenotype was rescued by constitutively active FMNL2, suggesting that Rac might normally activate the formin by opening up its autoinhibited conformation. Indeed, Rac1 bound to FMNL2's regulatory GTPase binding domain.
We then made use of a photoactivatable Rac1 so that we could activate this GTPase in time and space using blue light or a 488 nanometer laser. And indeed, at the moment we activate Rac1, not only Rac1, but also FMNL2 would rapidly, within 100 seconds or so, would actually go to the cell-cell contact site, and that would coincide with the appearance of LiveAct. So this was very nice real-time evidence for us that just by switching on Rac1, we could get the formin in its place and we could assemble the actin filaments.
In FMNL2 deficient cells, however, photoactivating Rac still induced the formation of lamellipodial protrusions, but neighbouring cells were unable to efficiently form cell-cell contacts. indicating that FMNL2 acts downstream of Rac to promote junctional actin assembly. One remaining question is how FMNL2's activity at nascent adhesions is coordinated with the function of the actin-nucleating ARP2/3 complex.
It's been shown for this formin that it is a poor nucleator but a very potent actin filament elongation factor. So if one would speculate further, what could be possible is that it elongates these filaments from up to 3 complex generated filaments, but this is something that needs to be addressed in the future.
Grosse and colleagues also want to investigate precisely how FMNL2 is recruited to adherens junctions, a process that requires the Formin C-terminal domain. In the meantime, however, you can learn more about how junctional actin assembly is mediated by Formin-like 2 downstream of Rac1 in the paper by Grikscheit et al., published in the May 11, 2015 issue of the Journal of Cell Biology.
You